Okay, we are good to go.
Thank you very much. So thanks a lot for coming for the second part. The first presenter is Ilya Lipkovich. He's an executive director at Eli Lilly and Company. He received his PhD in statistics from Virginia Tech in 2002 and has more than 20 years of statistical consulting experience in the pharmaceutical industry. He's an ASA fellow and published on subgroup identification in clinical data, analysis with missing data, and causal inference. Ilya, thank you very much for presenting in our workshop, and the floor is yours.
Thank you, Kostas, for a nice introduction and for inviting me. And this is an overview talk, modern approaches for evaluating treatment effect heterogeneity from clinical trials and observational data. And I want to acknowledge my co-authors on the recent paper, David Swenson, Bogdan Ratic, and Alex Dmitrienko. So I will talk about causal framework for heterogeneous treatment effects, outline for general approaches for estimating HCs, and what to look at in numerous papers on heterogeneous treatment effect evaluations. And of course, the topic of today's workshop is post-selection inference, so I'll talk more about it. And I want to start by advertising our recent tutorial in statistics in medicine on modern approaches for evaluating treatment effect heterogeneity, which is a follow-up of our earlier tutorial published in 2017. And so this talk is based on that recent tutorial. So I want to stress the complexity of estimating heterogeneous treatment effects as this intersection of three different activities is causal inference. And why it's causal inference? Because, say, for the linear outcome y, introducing two potential outcomes y1 and y0, if the same patient is treated with two different treatments, and you want to see at what's the treatment contrast given patient's covariates. And so that's obviously is causal inference problem. And I'll introduce it more formally the next slide. Now, X is possibly high dimensional. So of course there is machine learning and there is post-selection inference because there's so many different components. You need to select variables and models. And so there is obviously multiple hypothesis testing involved if you have multiple subgroups. So this intersection of these three components is very challenging. Now, to set up the causal elements here, so as I said before, there are two potential outcomes, assuming two treatments and only one is observed. So that's the fundamental problem of causal inference. And we want to introduce outcome function, which is essentially expectation of potential outcomes given covariates. And then we, for treatment, here, treatment indicator can be 0 and 1. And so we hope to estimate this using conditional average treatment effect, which essentially is conditional expectation of outcome given treatment and covariates. And that can be done also, obviously, under the assumption of treatment ignorability, which is trivially observed in randomized clinical trials. But in observational data, we hope that all the set of X includes all the confounders. And treatment contrast or conditional causal effect then is just simply the difference between this function M evaluated for patient treated and the same patient untreated. So in the studies with non-randomized treatment, we need to estimate propensity score, which is yet another challenge. And in randomized clinical trials, it's a known constant. So the literature on subgroup identification is diverse. And just a quick search on subgroup identification may give you several papers which span very different corners of this space. And so say that the first paper is by Vanderweel and Lutke and other people. They essentially look at estimating this conditional average treatment effect. And then the subgroups are defined essentially by thresholding them, giving different objectives. The second paper brings it into quite different context of interpretable subgroups, which is the tree-based subgroups, and that use constraint optimization motivated by the literature on individualized treatment regimens. The third paper is yet focusing on multiplicity control, and it finds subgroup as hypercubes, and the p-values are combined by Holmes procedure. So this is quite different type of research. And by the way, the follow-up of this research will be presented at the next talk on isotopic subgroup selection. So now our paper in 2017 tried to organize this enormously vast literature, putting like 4 different perspectives and or approaches. One is essentially estimating the outcome model. Then if you estimate it for covariates and treatment, so you can plug in treatment 1 and treatment 0, and then essentially you can tease out the treatment contrast, which is shown as a red line, delta X, from there. Now, other researchers focused on estimating directly delta X, obviating the need to estimate prognostic scores or emphasizing the direct estimation of these causal parameters. Yet the third group was focusing on directly estimating individualized treatment regimens. And so essentially we want to know what's the sign of delta X, not necessarily delta X, but the sign. Maybe you can estimate it directly without even estimating the conditional average treatment effect. And finally, there is a group of researchers who believe there is this magic subgroup in this covariance space, and they don't want to model all covariance space. They say we just model directly the subgroup using bump hunting on some other techniques or recursive partitioning, and that was termed the direct subgroup search literature. So things change a little bit, and there are a lot of interaction between these different groups of researchers. Here I want to emphasize a fundamental difference, which is not apparent for some naive readers of the literature on heterogeneous treatment effects. There is difference between the ultimate target could be the CATE, the conditional average treatment effect. This is a learner or meta learner, which essentially allows you to plug in X, and they give you prediction of delta for any future patient, as opposed to IT score, which is computed for a given data set, training data set, and it essentially requires not only X's but Y's. So it's a score. It's not the learner for the future patient. It's a score that you compute for a given patient, and the challenge is that for a given patient, even that you observe outcome in X, you don't observe 2 potential outcomes, so you somehow impute that potential outcome with the hopes that that estimated score and expectation give you the true delta, and we can call it pseudo outcome. And so there are many examples of IT scores, and one idea is just to impute or match this counterfactual that is unobserved and essentially which is shown here by tilde. And so for treated, you need to impute hypothetical control outcome under control. For untreated, you need to impute what would be if you treat it. Then you can compute these scores. Then another idea which was very popular is just you don't need to predict anything. You just form these inverse probability weighted scores. For randomized clinical trials, you don't even need to model propensity. You have known constant for propensity. So, and here you go, you can just model these pseudo outcomes and obtain consistent estimators of CATE. Well, unfortunately, as it was learned later, it's over-optimistic. The variability associated with these scores is so huge, the convergence rate so slow, probably wouldn't work unless you augment it somehow with actual prognostic models. And that's, you see augmented inverse probability scores, which are much more useful. And there is other transformations, notably there is Robinson's transformation, which I think was later also motivated by double machine learning, gave rise to R-learning. And here the idea is that, yes, you need to model propensity and outcome, the nuisance functions, but then you can combine it in a clever way so that even that the convergence is slow of these nuisance functions, your final estimation of CATE would go at almost parametric rate. So things change a lot from this over-optimistic hopes about just using simple transform outcome using inverse probability weighted scores to something much more sophisticated. So when you read in the literature on heterogeneous treatment effects, how you read the paper, there's so many papers and they, as I said, are very diverse. So I try to here mark certain important elements. For example, what is the method? Does it apply to randomized clinical trials only or to observational studies as well without with non-randomized treatments. And of course, it's an important interplay between confounders and modifiers of treatment effect. That makes model selection more challenging. Confounders are predictive of both treatment and outcome, and effect modifiers are only predictive of CADE of treatment contrast, and many methods cleverly combine estimating of both effect modifiers and prognostic effects in a clever way. So the number of predictors the procedure handles. Some methods just work with one continuous biomarker. That's enough for them, and they actually have very clever ideas how to do confidence bands, for example, for treatment effect given every value of these biomarkers. There are methods that would allow more covariates, and some of them even try to look at what happens if number of covariates increases as fast as sample size, and even larger than sample size. So model complexity. What's complexity of the model space where the subgroups reside? And it could be the subgroups defined based on black box functions of covariates. So you estimate CADE and it's high dimensional, then you threshold it. And that's one approach. Another approach is that you directly want to find these biomarker signatures, which are interpretable. And that's sort of another type of model space. Now, there's strategies combining this two. And a very popular idea would be, for example, to compute IT score delta using all these approaches that I outlined before, and then say using doubly robust scores. And that already involves fitting different nuisance functions using machine learning. And then you apply card tree to the student outcomes and to prune the tree. And so it's kind of sometimes hard to understand what is the complexity of the model and how the model complexity is controlled to prevent data overfitting at different steps. Now, what is important to understand is that optimal tuning at each step doesn't guarantee optimal estimation of the target causal estimates of the target parameters. Now, what is the output produced by the method? It may be individualized treatment contrast, which I already outlined and focused on before. Now, biomarker signatures of promising subgroups. For example, one biomarker less than threshold cutoff and another greater. Optimal individualized treatment assignment rule, which is, say, if this delta for a given patient exceeds some threshold, maybe 0, then treat. Otherwise, not treat. And it could be something else, but that's another popular output. And predictive biomarkers, maybe you just want to output which biomarkers are effect modifiers using variable importance score. So what inference is done, if at all? And a lot of papers don't offer inference. They just say how we estimate something. Inference on presence of heterogeneous treatment effect, inference on CADE, inference on subpopulations, and could be controlling the probability of selecting the right subgroup, or it could be that you're just giving whatever subgroup you found, you want to estimate the true treatment effect contained within this subgroup. Inference on individualized treatment roles. And there's a concept of value of individualized treatment role. So what if this rule applied to all population? And what that's potential outcome given that treatment returned by that rule applied to all data, what's expected value? And inference on selected predictive biomarkers. So for example, Kostas presented this method of knockoffs that controls false discovery rate on predictive biomarkers. Now, inference on presence of, now I'm going quickly through different methods. So inference on presence of heterogeneous treatment effect. It's a difficult problem, and there are several approaches. One is advocated by Chernozhukov and further extended by Effie and Wager. So essentially, you get some estimate of delta x, some learner for CADE. Now you project it in the linear space in Y, and here is important that you use some kind of cross-fitting versions of these outcome functions, propensity functions, and this delta. So essentially, for each patient, you obtain the estimate without that patient fitted in that model, and there are different versions of that. So now you can prove that this beta equals 1 would show that delta is indeed well calibrated, and the inference on the significance of the beta would actually be the inference on over heterogeneity of treatment effect. Other approach advocated also by Chernozhukov and then extended by Imai and Lee in the different procedure is also use cross-validation, cross-fitting, but it is divided this estimated delta into groups that are representing monotonically increasing treatment effect, and then you essentially test the hypothesis that all these true Kate within these groups induced by estimated delta are the same or not, and it's implemented in package eval ITR. They derived asymptotic variance for the test statistic under cross-fitting framework, which is nice, and it's assuming treatments are randomized, so it doesn't assume that you have non-observational, a non-randomized trial. Now, there are other ideas, for example, variation of Kate over covariate space, which is essentially variance of this Kate over covariate space, and then Levy colleagues developed cross-validated TML estimator with simultaneous inference for average treatment effect and this variation of Kate. So inference on delta X on the Kate. This is also a problem which is quite difficult. The point-wise confidence intervals were obtained often using post-selection inference on penalized regression that you used to model outcome with treatment by covariate interaction. That was approached by Ballerini. And then if you include treatment by covariate interactions, you hope they, sufficiently modeling the outcome and the heterogeneous treatment effect, you can use post-selection inference on Lasso to tease out the confidence intervals for delta for Kate for every X. And there are other approaches, causal random forest, which combine our learning with innovative ideas on bagging estimators and inference for bagging estimators. Now, simultaneous confidence bands on delta X, it's another approach that was taken by researchers. Here, it was important, for example, an approach by Lee, that you do it in two stages. And first stage, you do high-dimensional modeling of all nuisance functions to compute these doubly robust IT scores, hoping that it will converge at parametric rate to the true scores. And then in second stage, you use a much smaller number of candidate effect modifiers to model Kate by regressing these scores on the smaller set, because you think that it's well known and it's just a few covariates that could be effect modifiers. Then they argued that you can ignore uncertainty induced by the first stage if the convergence is fast enough at parametric rate, and they propose these non-parametric kernel estimators. There are Bayesian approaches that extend BART, and I wouldn't talk much about it. These Bayesian approaches now motivated inference on identified subgroups. And the first type of inference would be controlling the probability of selecting the right subgroup, which was developed by Schnell and colleagues. And here you essentially threshold these Bayesian estimators of delta with, say, some threshold maybe at 0. And that's the true subgroup which you unknown, but you create bounding subgroups, lower and upper subgroups that guarantee that the true subgroup is contained with given probability. And so one is called exclusive set, another inclusive, and it's essentially based on thresholding the lower and upper limits from the credible sets for the Kate. So if your exclusive set is empty, that suggests, okay, there is no heterogeneity. Now, placing guarantee on a set of subjects suggests testing for positive treatment effect at individual patient level, which actually is idea that is pursued of the last talk today, interactive identification of individuals with positive treatment effect. And it's an interesting concept that you essentially test Schwab hypothesis. It comes from the Fisher's framework, but how you interpret the subgroup of patients that are identified of having positive treatment effect if they are becoming kind of removed from the covariate space. It's kind of hard to link it with particular biomarker signatures, and that's a challenge, I think, for interpretation. Another type of inference of subgroup is, okay, we believe our subgroup is good. Now, given estimated subgroup, what's the honest treatment effect? And there are Bayesian shrinkage method. And frequentist resampling methods. You can contrast between the wild machine learning algorithms, which essentially used to compute the bias for over-optimism bias for treatment effect estimated naively by resubstitution of the identified subgroup in the same data. Now you can apply it to bootstrap data, but the challenge is application of the same machine learning methods to bootstrap data would give you quite different set of subgroups. It's hard to link them, and other type of methods are more tame. They sort of they consider a well-defined set of subgroups defined by, say, by thresholding one or several biomarkers. Then for each bootstrap sample, you can clearly remap them. There's a clever methods that will adjust for the maximum selected subgroup or best subgroup selected from this relatively small set of well-defined subgroup. And combining the two frameworks, for example, throwing a lot of interactions in lasso and use debias lasso, plus bootstrap adjustment on those subgroup in space in terms of these interaction terms, also pursued by some researchers, is essentially a combination of these two. And like, is it possible to do inference on data-driven subgroups without resampling or test data? It seems impossible, but there are indeed approaches that are using the idea of masking some aspects of the data. And so, for example, Jason Shu and other colleagues, they argued that, say, you could estimate scores, say, by matching patients, and then these IT scores could be used to identify subgroups, ignoring the sign of the score, using absolute values. They argued that the chances that you find out subgroups where patients with very high positive and very high negative scores will be combined very small under certain reasonable assumptions. Then, because you don't use the sign of this IT, you can use essentially the known distribution of the sign of delta under null is 0.5. And then you can construct Taiwan error procedures with strong Taiwan error control or false discovery rate control on the set of groups under null. And finally, inference on individual estimated regimens, and it's also very challenging. It's non-regular problem. It's non-smooth because different regimens can give you the same value, even if you have one single stage of estimating ITRs. Now, one should consider estimating or inference for the value of estimated ITR and the inference for the value of the true underlying unattainable regimen. And so there are indeed procedures that guarantee that the confidence interval obtained for the estimate ITR would also cover the true ITR, which I wouldn't go into that. And there is a very interesting approach proposed by Imai and Lee using the cross-fitting and again cross-validation framework to estimate what they call population average prescriptive effect, PAPE. And the idea here is that they want to control the proportion of patients who would be assigned to treated, which is a budget, and given that budget, they would consider different procedures to allocate the same number of patients to treated, arguing that not controlling that in computing just the value of any ITR would be unfair. So essentially, you compare the regimen with a similar regimen or maybe random regimen. Here, the red highlighted red is the value if you randomly assign the proportion P of patients to treated. And then they develop inference on that. And then finally, I want to advertise our site where we update regularly and we put all these links to different procedures. To summarize, the shift from ad hoc subgroup chasing methods towards principal methods of personalized precision medicine, utilizing ideas from causal inference, machine learning, and multiple testing emerged in the last 10 years, producing a vast number of diverse approaches. Naive multi-stage methods requiring fitting response surface regularization. For them, regularization bias can be large as each step is optimized for prediction rather than for the final causal target. Doubly robust strategies for CADE are preferred, and post-selection inference is challenging. I reviewed some recent methods, mostly within frequentist domain. Here are some references. Thank you.
Thank you very much, Ilya. Very nice talk. Now, because we ran out of time, we can take a couple of questions. Are there any questions? If there are not, maybe I can ask one question about you said about the inference of the presence of the heterogeneous treatment effect that there are around 3 different methodologies. Did you compare them? Do you have any intuition about them? Did you have some comparison in the paper?
Because which 3 methodologies?
So, for example, the best linear projection, and then you have the variance. The variance also the Kate for the last one. I was wondering if you had some. Do you have
I didn't do it, and maybe that would be good next step. In our review paper, we didn't focus on how good is the inference. We compared the bias and mean squared error just of Kate. So we kind of focus on simulating challenging scenarios and just comparing different meta learners that estimate Kate, but we didn't actually compare the methods that actually provide inference and how well they, for example, confidence interval cover that they provide cover the truth, what's the nominal rate. And I think that could be the next step for us.
Yeah, thanks. Yeah, it's great. It was a great review. And any questions? So I saw also someone post in the chat maybe the web page. So let's move. Thank you very much, Ilya. Do you want to start sharing? Yeah. So we'll move to the next presenter. It's Manuel. Manuel receives a master's in statistics from University of Vienna before joining the University of Cambridge for his PhD. There, he's jointly supervised by Richard Samworth and Henry Ripp from the University of Bristol. The focus of his research so far has been on developing methodologies for the identification of subgroups under strong type 1 error rate guarantees. And actually, we're in the very lucky position of working with Manuel the last 2 months of examining how these methods can be applied in clinical trial data. And more generally, he's interested in structural multiple testing and finite sample assumption and finite sample assumption inference. So Manuel, the floor is yours.
Perfect. Yes. Thank you for.
Maybe.
I am muted. Maybe you have to turn down the speaker.
Let me.
The room speaker.
Now.
Let's see. Yeah, that looks good. Okay. Perfect. Yes. So thank you for the introduction, and also thank you to Costas, Frank, Dominic, Vivian, and everybody else involved in putting together what has already been a fantastic event. And yes, as Costas just mentioned, so I'm going to talk about isotonic subgroup selection, which is work that I have done with these 3 fantastic people, and this work that has recently been accepted at JRSSB. And so let's start off with a very basic question. What is a subgroup? What should we think about when I talk about the subgroup? So let's start off with the most basic setting one could think about. Say you have the data from some patients, as in the scatter plot here, and for each patient you record some response and some biomarker value. And then the most simplistic approach to analyzing subgroups you could think about is to do some pre-selected split of your biomarker space, and then look at the biomarker negative and the biomarker positive patient group, and then look at something like their average group specific effect. Now, starting off with the same data, I want you to think about things in kind of the other way around. I want you to think about, first and foremost, about having selected a threshold, like a target response. And then what you want to do, so there is some true data generating mechanism going on in the background here, and this is unobservable. I'm going to talk about this in terms of a regression function or population regression function, but just to make things clear, whenever I say regression function, I mean a true data generating mechanism. The method I'm going to talk about is not fitting any kind of regression function or something like that. So regression function means the true data generating mechanism. And this gives the expected response conditional on the biomarker value. And you can see here that at some point, this expected value, this regression function, is crossing the target response, namely if the biomarker value exceeds a value of 0.8. And this is the notion of subgroup I am thinking about in this talk. Now, to set things up a bit more formally, So what we have is we have some data. We observe independent and identically distributed covariate response pairs. The covariates can be arbitrary dimensional. Here it's a univariate example, but they can be d-dimensional. And then what the user is specifying is, first of all, the threshold, which is kind of application driven, and then a maximal type 1 error rate. And why do we need a maximal type 1 error rate? Because here you can see that there is a point, I've just discussed this, at the point 0.8, where the regression function starts to cross the target response level. But of course, we can't observe this. We don't know the black dotted line. We only see the blue data points. And so in kind of clinical terms, we would call everybody with a biomarker value larger than 0.8 a super responder, for instance. And when we try to establish this unknown cutoff here, there are two mistakes we can make. The first one is that we set it a bit too loose, to choose too small a cutoff, and claim that more people are super responders than actually are super responders. And this is what we term a type 1 error in this setting. The other error that one might make is to choose the group too restrictive, to not select everybody who is a super responder as a super responder. And this we are kind of fine with in terms of type 1 error guarantee. So this would be this would not be a type 1 error to choose the biomarker as you see it here, to choose the biomarker cutoff as you see it here. And so the task that I'm focusing on in this talk is to identify a subset A hat of the covariate domain such that for each and every single value within the subset, we have a regression function value larger or equal to tau with probability at least 1 minus alpha. And this is our type 1 error rate control. And subject to this main constraint, we want the subset, the selected set to be as large as possible. And this we will quantify by something I'll call power, which is the probability of if you observe now a super responder, a patient whose expected effect is larger than tau, what is the probability that they lie in the selected set? And so if you focus your attention to the plot to the right again, this again is perfectly fine in terms of type 1 error, but selecting smaller biomarker cutoff or an even smaller biomarker cutoff would be even better in terms of power. And sadly, if we want to control type 1 error over all possible distributions, we will inevitably have trivial power. So we will need to make some assumptions to make this problem feasible. But before I talk about the assumptions, let's look at one specific example that I've been focusing on during my time here at Novartis over the summer. So here we looked at a phase 2 study with about 250 patients who received a new drug with varying dose levels, and some patients faced adverse events. And so the question is, can we predict which patients are at risk of adverse events? And we apply subgroup selection here in the following sense. We transform the responses by saying that the response takes value equal to 1 if a patient stays adverse event free and 0 otherwise. And then A hat only contains those covariate configurations, those patient characteristics for which the probability of staying adverse event free exceeds a certain threshold. So for instance, 0.95, if you want to be 95% sure that the patient does not face any adverse events. And the decision process, this kind of very strong type 1 error guarantee allows you to then follow through on, is that once you have computed your set A hat and you observe a new patient X, you can check whether X falls into your selected subgroup A hat. And if so, you can be reasonably sure that the patient is not going to face any adverse events, since the patient's probability of not facing them is above the threshold tau that you've chosen. If on the other side, X falls outside of your subset, then you essentially don't know anything about them. We don't provide any estimates for either. We only tell you whether eta is above tau or whether we don't know if it's above tau or not. One thing that should also maybe be mentioned here, the very restrictive notion of type 1 error guarantee that we've chosen here also allows you to follow this decision process if your clinical population, which you might base the estimate A hat, differs from the real world population on which you then apply this decision process. Okay, now let's look at the specific assumptions that we have been looking at in the isotonic subgroup selection paper. So first of all, we assume that the true population regression function is increasing, as in the example before. And what this means in multiple dimensions is that if you have 2 points like x0 and x1, and x0 is smaller in each and every single coordinate than x1, so in the plot to the right, you see x0 lies to the lower left in the lower left region of x1, then its regression function value must also be smaller than that of x1. Or conversely, you know that all points to the upper right of any point x0 need to have larger regression function values. And then the second condition that we have is one on the error distribution of your responses. So the responses need to be either homoscedastic Gaussian, bounded in some known range, or sub-Gaussian with known variance parameter sigma squared. And if you don't know what sub-Gaussian means, don't worry about it. Replace sub-Gaussian every time I say it with the word Gaussian in your head, and everything to follow will still make sense. And in the very recent literature, there are 2 other approaches that have been examined. So one depends on Hölder smoothness of the regression function by my collaborators. This is what we have heard about also in the previous talk. And the other literature on this assumes the suitability of a generalized linear model. And this is work by Fang Wan, Wei Liu, and Frank Wetz. Okay. As for the high-level strategy of our procedure, so once again, just to emphasize, our procedure, even though it has isotonic in the name, does not fit any isotonic regression function. We're not fitting any model in any way at all. The only thing that we do is look at localized null hypotheses of whether at certain points in our covariate domain, the regression function lies below the threshold tau. So these might be some patients that you observe and their covariate configurations. And what we do is in the first step, we subsample M out of these N patients. We can also set N equal to N, and everything I say will follow through. Then for each of these N points, we calculate a p-value for this localized null hypothesis. And then we apply to these p-values a multiple testing procedure with family-wise error rate control. And for instance, we might reject the hypotheses belonging to the blue points here in this plot. And then because of the monotonicity assumption that we made before, we know that if at those blue dots, the regression function takes a value larger than tau, at all points to the upper right of any one of these points, we also need to have regression function values larger than tau. So we can select everything that's here in the gray area. Okay, I'm not going to say more about the first step. Regarding the second step, the construction of the p-values, this alone is actually not trivial under the assumptions that we have made, especially since we are interested in finite sample validity. And so let's say you're interested in the point x0 equal to x7 here in the middle. If you're not willing to fit any kind of model to your data, the only points that you can use in order to reject the null hypothesis there are the points that lie to the lower left of the point x0. And so the issue is we don't know how many of any of these points we should be including in our test statistic. So we start with the point x0 itself. We look at the concomitant response, and we do this usual set transformation essentially. And we record this amount of evidence that this one point gives us. And then we look at the next closest point in subnorm, for instance, but the kind of distance doesn't matter. We look at the set score from that next closest point, and we add that evidence onto the previous evidence. Then we go to the next closest point and do the same thing. And we keep doing that. And by doing so, we construct an online or sequential procedure, because this blue line, what it gives us is something that's called a martingale, which under the null hypothesis should have a downward trend or at best stay roughly constant. But if it has an upward trend, that is an indication that the responses are larger than it should be under the null hypothesis. And so what we can do is we can look into the literature and use absolutely fantastic work by Aditya Randas and his co-authors, such as in these references, where we have time uniform boundaries that this blue martingale should never cross beyond. And so we can invert these boundaries in order to construct a p-value for this null hypothesis without any kind of model fitting. So this is how we calculate p-values. As for the multiple testing procedure, I don't unfortunately have time, even though it would be a lot of fun to do. I don't unfortunately have time to go into all the details here, but just let me say, so we are only focusing on the M subsampled test points here, kind of. And we've just seen how we can calculate the p-value for x7, and we do the same thing for all N test points. And then the nice thing here is that these local null hypotheses they follow a logical structure. Some of them implicate others. And so what we do is something that's very closely related. So we do something that is an instance of the sequential rejection principle, and it's closely related to the work Frank Ratz has done 15 years ago, where we take the alpha budget and we allocate it in a smart manner within our hypotheses and do localized comparisons between p-values and the local alpha budgets. And each time we make a rejection, we get to recycle some of the alpha budget. And we do so in such a way that we start in the upper right corner and move further and further down to the lower left, where it will be harder and harder to make rejections. So this is how the procedure works. And the resulting set I'll denote by A hat ISS for isotonic subgroup selection. And our theory tells us that we have this finite sample, very strong type 1 error guarantee, even conditionally on the covariates that we observe. So we can also use this for any kind of fixed design setting. And we also have some results showing that we can even have some violation of the monotonicity assumptions that we made. In particular, what we can also have is that the regression function is monotonically decreasing. So for increasing, we just have to flip some signs, and the same exact procedure follows through. And then we also have theory telling us that this procedure that we are proposing is minimax optimal for those that are interested in these power guarantees. Okay, now let's look back at the first application that I've looked at during my time here at Novartis. This is the one I've already teasered in the beginning, where we look at predicting adverse events and patients at risk of adverse events. And the way we set up the simulations here is we use the package SynPop to resample from the exposure distribution that we have in the clinical trial. So to start with, we look at the univariate example for illustration purposes mainly. And then what I did is, to the original data, I fit 4 different regression functions. One is based on logistic regression, so we have a logit or inverse logit curve here. Then in the second panel, you see the same thing, but with a highly non-monotonic component. In the third plot, you see essentially the same as the first plot, only that we truncate the function so that there is actually no subgroup in our definition here. There are no like super responders or anything like that here. We never trust above the thresholds. We shouldn't select anything here. And the fourth is one where we have kind of a monotone decrease, but it is a bit squiggly, and it's kind of there are minor violations. And just to re-emphasize, these black lines we then use to sample our responses, but these are not part of our method. They are only part of the data generating process, and then our method takes as input the blue dots, the red lines, and does this localized p-values with multiple testing procedure. And for our results, we compare our method to the parametric method that assumes a GLN by Feng Wen and Korfas that I've mentioned before. And if we look at the type 1 error rates, as we vary the sample size in these 4 settings, you can see that except for the case where we have a highly non-monotonic component, ISS nicely controls the type 1 error rate by never crossing above this red line here, which tells us the alpha. And it's kind of much better at controlling type 1 error rate in these settings compared to the parametric method. But of course, nothing's for free. So we see an effect here that we have, of course, also less power than the parametric method. And note the third panel here is empty, of course, because there just is no subset that we should be selecting. And then another thing we looked at, so just to illustrate very briefly that we can also do something in multiple dimensions. So here, the solid lines are the same we've seen in the previous slide, and the dashed lines are if we also include a binary covariate. So we can also handle categorical variables with our approach. Okay, and then the second application we looked at concerns heterogeneous treatment effects. So here we use the package BenchTM by Sophie Sun and a lot of fantastic people here at Novartis, which is designed specifically to simulate data inspired by clinical trials with treatment effect heterogeneity. And then we've already heard a lot about this in the previous talk, so I can hopefully go through it a bit quicker. So we have this conditional average treatment effect, and this is what we will target in our setting. And specifically, if we have some efficacy threshold, that could be a clinically significant threshold, but it could also just be equal to 0. We want to identify patients whose K is beyond tau. So if tau is equal to 0, you maybe are just interested in making a non-inferiority claim here. And the difficulty is, of course, the fundamental difficulty of causal inference is that we only observe one or the other potential outcome. And so this is kind of purely empirical. What we did here, we don't have any theory in this yet, but what we then looked at is using this double robust machine learning framework in order to generate pseudo observations mimicking the individualized treatment effect scores. And so a bit more about the simulation setup. So again, we use this BenchDMR package, and the responses are standard normally distributed either around G0 in the control arm or G0 plus K in the treatment arm. So this first column here gives you the K, which is what we target. And in these 4 settings, in order, we first have an increase similar to the CDF of a Gaussian distribution. Then we have a linear increase in the K. Then we have a K driven by an AND condition between 2 properties of the covariates. And then we have an OR condition. And if we look at, and then we apply our method based on either as an oracle setting where we observe the individualized treatment effect, or where we have to resort to using these pseudo observations. And so if we look at the results, we see something that's pretty much in line with what we have seen in the previous application, where ISS neatly controls the type 1 error control. You can see that whether you use the pseudo observations or the true observable individualized treatment effect, there isn't too big a difference here. We do, regarding the power, we see some decrease in power when we use the pseudo observations, but there might also be a lot of room for improvement in terms of what kind of learners we use and things like that here. And with that, to wrap up, just some extensions that are in the paper. First of all, there in more detail, it's described how we use adaptation to unknown variants of Gaussian errors, which is based on the universal inference paper by, again, Aditya Ramdas and Korthus. And then we also talk about bounded responses, such as in classification more there. And then we also have something for heavy tails for isotonic quantum regression. And we have extensive simulations and further applications. And there is also an R package called ISS on Tran. So please do feel free to try it and let me know what you think about the method. And so to sum up, just 3 short take-home messages. So first of all, subgroup selection with strong type 1 error guarantees is possible in the isotonic regression setting. And so just think about that. What we can do here is we can have confirmatory type, strong type 1 error guarantees when we, at the same time, without data splitting or anything like that, identify subgroups of interest and also make a statement about the effect within this subgroup. And our method specifically is computationally feasible and minimax optimal up to polar logarithmic factors. And in common situations, you can apply this without needing to know any kind of smoothing parameters or nuisance parameters that would be unknown in practice. And here are some of the key references. And with that, thank you all, and thank you again to the organizers.
Thank you very much, Manuel. There is already one question in the chat by Aditya. The restriction to the sub-Gaussian case is the restriction just for illustration in the talk? If not, what's the bottleneck in going beyond the sub-Gaussian case? Because those time uniform boundaries certainly exist for more general tails.
Yes, exactly. Right. And so that's an excellent point. Thank you. So our approach, as you could tell from the presentation, is very modular. We have the 2 steps, one where we construct the p-value, and then when we have the multiple testing procedure where we try to leverage the logical structure that we have between hypotheses. And so for the first part, for the p-value construction, you could, of course, also work with any other kind of distributional assumptions. And so I know that your work covers a wide range of different distributions. In our paper, we didn't cover all of them, but you could certainly, I think for many of them, probably very easily construct p-values for other distributions by, again, looking at your work with Howard and others. Yeah.
Are there any other questions? Yeah, Asher, do you want to unmute? Now I need to.
Yeah, thanks so much for the great talk. Can you hear me? Sorry.
Yes.
Okay, I was just wondering. So, do you have intuition about sort of the best way to choose this subsample size M? And like, do you think it's really sensitive to that? That's sort of my question.
Right. For the most part, it isn't. So there are some reasons why you might want to choose n smaller than n. So one is that in the multiple testing procedure, you test m hypotheses. And so any multiple testing procedure will kind of have some decrease in power, I guess, if you increase n. So it's just harder to control the multiplicity the more hypotheses you have. And so especially if you're in a small n size, maybe having then n equal to n, you might be losing too much for this multiplicity correction there. Another thing is that the multiple testing procedure, so what I've showed you in terms of computational efficiency, for reasonable sample sizes and small dimensionality, the procedure is extremely quick, but the runtime increases the higher dimensional it is. And so there might also be, and it also increases with m. So also for computational reasons, maybe you want to choose m smaller. But if I would have to give a recommendation to anybody trying to apply this method now out of the box, I would say choose m equal to n. This is also the default option in the R package, because our power guarantees tell you that the effect of n is much stronger than the effect of m on the power. So yeah.
Thanks so much.
Thank you.
Thank you, Manuel. And we already have Aditya. Sorry for running a bit late. Now, the next, last but definitely not least, is Aditya Ramdas. He's an associate professor at Carnegie Mellon University in the Department of Statistics and Machine Learning. He received the Sloan Fellowship in Mathematics, the IMS Peter Hall Early Career Award, the COPSS Emerging Leader Award, the Bernoulli New Researcher Award, an NSF Career Award, an Adobe Faculty Research Award, and Google Research Scholar Award. His main research interests include selective inference, game theoretic statistics, and modern predictive inferences. Now, thanks a lot, Aditya, and yeah, we're looking forward to the presentation. The floor is yours.
Thanks a lot for the invitation and for organizing a wonderful session. All of the other talks have been excellent as well. Hopefully you can see my screen and hear me well and so on. If not, please let me know if it gets stuck at some point.
Everything is great, thanks.
Perfect. So I'll be talking about, well, the title here, interactive identification of individuals that have a positive treatment effect while controlling the false discovery rate. Before I get into the details of this paper, I want to just give a little bit of high-level overview of this area of interactive multiple testing. Okay, so data scientists want to interact with their data and use their intuition, use their priors in order to exploit structure in the data. However, many classical statistical methods were not built to handle interactivity with the data because of double dipping and selection bias. And so there's a need for principled statistical methods that work in kind of sequential and interactive settings. So here's like a hypothetical data scientist wish list. They want to use their prior knowledge and intuition about the problem. They want to incorporate structure in the form of soft or hard constraints. They would like to interactively play with the data rather than just throw it into a software and get an answer and stop. So they would like to somehow participate in the process, a human in the loop. They would like to employ flexible probabilistic modeling tools, and they would like to be potentially robust to unknown dependence between outcomes, covariates, things of that kind. And the challenge, of course, is while wanting all of these things is how do you get correct statistical inference? And this talk will talk about some progress within multiple testing and causal inference to do this. So interactive testing, essentially, again, this is a high-level flowchart, but it's going to allow loops. So you have an initial data set that we have collected. We're not going to show the whole data set to the user, to the statistician. We're going to mask the data in some form and only show part of the data to the statistician. And the interactive test is going to be a multi-step procedure where if you're not ready to stop yet, or we have not rejected the null, or something of that kind, if some criterion is not satisfied, then we progressively unmask the data. So we've hidden some part of the data, and we progressively show that hidden part to the statistician. Statistician is allowed to mull over what new thing they've learned and change what they're doing at the next step. And so there's a loop over there where they get to interact. And then at some point you can stop and hopefully reach your desired conclusion. So this is going to kind of be what we're going to talk about today, how to construct such things. So And the key part is that the test itself can be customized and revised after observing the data. So you don't have to kind of pre-commit to a particular model class in advance and so on. So again, zooming out a bit, some background, a timeline of interactive multiple testing. So I think, according to me, there was a seminal paper by Lay and Fithian in 2018, which conceptualized this idea of masking and designed what's called the ADAPT algorithm for interactive FDR control. That was a JRSSB paper 6 years ago. Soon after, I mean, I joined forces with them, and in a Biometrica paper, we generalized this idea of masking to other masking functions and showed how to actually enforce certain structure that you might have in advance. And then my PhD student, Boyan Duan, got involved in this line of work, and she graduated a few years ago and is currently at Google. But she started to take this forward to other problems other than interactive FDR control. So, for example, interactive tests for the global null, interactive family-wise error rate control, interactive Wilcoxon and Friedman type tests. And this is what I'll talk about today. Is kind of most of these past works they were focused on somehow p-values, like or that was the underlying thing that we were trying to mask off and things of that kind. And so in this talk we'll kind of move away from the setting of p-values, and I'll show how to control like the false discovery rate in a causal setup where we don't have p-values and yet have this kind of interactivity. And then this has kind of culminated with a recent this year's JASA discussion paper, which proposes a different way of promoting that interaction using something called data fission, which now allows you to do beyond FDR control, but also estimate treatment effect sizes and get false coverage rate control and things of that kind. But my talk is essentially going to be on this earlier work before this year's discussion paper. Okay. So here's the title of that paper again. So it's Interactive Identification of Individuals with Positive Treatment Effect While Controlling FDR. This is published at the Journal of Causal Inference this year, and it's joint work with Boyan, and who was co-advised by me and Larry. And that's the archive identifier for quick reference. Okay, so here's a visual of what we're going to be aiming for. It is a weird goal, and so it's maybe good to try and understand what the goal is with a simple visual example. So let's say we're given, we have some covariates for these people, age, weight, some things of that kind. And here I'm just plotting two of the covariates. So let's just say that we only have two covariates, and they're just plotted on the unit cube for simplicity. I'm going to start out with a simple randomized setup to describe the problem and procedure, and then at the end I'll tell you that in the paper has extensions to going even to observational settings and so on. But for now, let's take IID Bernoulli half treatment assignments. And so what you're seeing on the color of each of these plots is the purple people were treated and the orange diamonds were control. Okay, so these were the treatments. The color is the treatment, and the shape also is the treatment assignment. So every point on this plot that you're seeing is a person, and half of them roughly were treatment and half were control. Okay, we have some potential outcomes for the treatment and control. These are YI control and YI treatment. Obviously, we never observe both. We observe one of them depending on what we assign to the person. And so what we do observe, we're going to assume consistency that YI, the thing we observe, is going to be YI treatment if I assign them treatment, and it's YI control if I assign them control. And so this is on the right plot, maybe the observed data. So, X1, X2 again on the same axes, but now I'm, instead of plotting the treatment assignments for the same people, I'm plotting the observed outcomes. Okay, and so the small outcomes at the bottom left, large outcomes on the top right. Okay, so this is the data set. So that's the data set that we have. And the question that we're asking is, once you have access to this data set, which individuals, which subjects have a positive treatment effect? So I'm asking which of these people here have YI treatment greater than YI control. So I'm asking for individual level inference. So this is again a weird question, and it will continue to be weird as I go along, because unlike, for example, like several of the previous talks, say, for example, when you have the conditional average treatment effect and you're thresholding at some level, you can imagine maybe that if I gave you an infinite amount of data, you could actually learn the conditional average treatment effect reasonably well and so on. But the difference over here is we will never be able to actually verify which individuals have positive treatment. No matter how much data I give you, you never see both YI treatment and YI control. And so in some sense, even in the limit of infinite data, this question somehow seems like, how could I possibly answer this question? I only observe one of the two. But this is the question we're going to try to answer. Okay, so if we just, for simplicity, look at the person at the top right, they were they were treated, and we see that they have a large outcome. And you can ask, well, did they have a positive treatment effect or not? And looking somewhere else, here are two people kind of close to each other. One was treated, one was control. You can see some difference in their outcomes, and you can ask, well, were these people, do they have a positive treatment effect or not? But we want to know something about every one of these individuals in some sense. Okay, so here is the actual answer. Since this was a simulation, I'm telling you the answer. So in this particular case, the blue people in the middle, those are the people that had a positive treatment effect, and everybody else on the boundary did not. Okay, so so the goal is to identify these people, and we're going to output a set R in the end as our claimed people that we think have a positive treatment effect. And so that's the blue box. Let's say we proclaim this set. Of course, we don't get to see the middle box. And in that set that we've proclaimed, the red area has the false discoveries. I mean, again, we don't know these, but now that I've shown you the simulation truth, ground truth, you know that the red box is the false discoveries. And so we want this error control on this expected proportion of false identification. So we want to control the expected fraction of the people that have YI treatment being less than YI control among the people that I've identified in the blue box, divided by the size of the blue box. So this sounds a lot like an FDR controlled multiple testing problem, but in the language of causal inference, there seems to be some issues around can I even solve this, because I'm not given p-values, I'm given these types of observations. How do I actually test these types of hypotheses? Okay, so we actually chose to formalize the problem of identifying individuals using the language of hypothesis testing, but I don't think this goal of FDR control has been identified before as something to target. So here's one example of a formal problem setup. So you could define the potential outcomes as random variables. So you could say, for example, that the null is that the distribution of YI treatment given Xi is equal to the distribution of YI control given Xi. So for the ith person, so this is the ith null hypothesis. So for the ith person, the distribution over those two potential outcomes are equal. And for the alternative, this is just an example. You don't really need a strict definition of an alternative, but you can think something like there's some kind of a stochastic dominance that YI treatment should be stochastically larger than YI control under the alternative, something for that particular individual. So you have n people and you have n null hypotheses, one for each person, and those distribution could all be different. Okay, so they're conditional distribution, so they could be different for each person and so on. So now there's nothing particular about modeling the potential outcomes as random variables that's important at all. So in the paper, we describe alternative definitions where you could treat the potential outcomes and covariates as fixed. So that's not going to be critical. I'm just giving you an example of how what a reasonable null hypothesis would be. And again, just to point out, in the paper, we show how you can extend this to error control for non-positive effects where under the null, the distribution is stochastically smaller than treatment is smaller than control. But for simplicity, let's stick to the testing zero effect against positive effect for intuition. Okay, and for most of this talk, I'll just deal with randomized experiments without interference. So I'm just going to assign treatment at random with probability half, treatment control probability half, and conditional on the covariates, the outcome of one subject YI is independent of the assignment to any other subject AJ. Standard, no interference, randomized experiment. Okay, so I've run a randomized experiment. How can I identify which individuals have a positive treatment effect? Okay, so we want to control this false discovery rate as I've described before, and the output of our procedure I call the procedure I cubed, interactive identification of individuals. So the output of I cubed will be the set R with a provable guarantee that the FDR will be small and hopefully reasonably high power, like in practice, in experiments, things like that. You cannot have a two-sided guarantee. Of course, I can have the alternative be so close to the null that essentially you get FDR control but no power. But the hope is that if there's heterogeneous treatment effects in the population, even if the average treatment effect across the whole population is small, there might still be individuals who are, as mentioned before, like who are super responders or something like that. And the question is, can we identify these people? Okay, and so again, this in the setup of the experiment that I showed you, this is what our iCube produces. So the green set of people is what is the output from I cubed. And you can see it's, well, this experiment in this, it's reasonable. You have reasonable power. You can see some false discoveries kind of around, scattered around, but it's not too bad. But I guess one of the most important points is you can identify subjects, regardless of whether they were treated or not. So, I mean, if you actually look at that box, like there's several people who actually were assigned control, but we're still able to determine that, well, if they had been assigned treatment, they would have had a higher potential outcome, despite not having observed their potential outcome under treatment. So that's interesting. So you don't identify only treated people. You could be identifying people who were controls. Okay, so now let me tell you more about the algorithm itself. Like what is this doing? How do I get to that green curve, the green identified set? Okay, so it's going to be interactive. So I have to describe this a little bit slowly. It's a little bit odd. So what we're going to do is we're going to start with the initial set being everybody, and say maybe everybody has a positive treatment effect, right? So R0 is a hypothesized set. And we're going to give the statistician initially not all the data. Okay, so we're going to give them only the covariates and the outcomes, but not the treatment assignments. Okay, so the explorer, which is the statistician, has yi and xi. And the oracle, which is your computer essentially, has AI, which is, the statistician is not allowed to initially see the actual treatment assignments. Okay, so the algorithm works as follows. So now y hat is going to be just any estimator of expected value of y given x. So you just essentially run a regression of y on x on your given data set using any model, and compute residuals EI, which is like yi minus your fitted y hat of xi. Okay, so EI is going to be the errors or the residuals. And now what's going to happen is at this step, the statistician is going to just somehow give the oracle these EIs, and the oracle, because they know the AIs, can compute a treatment effect estimator for each individual. So delta hat I is an estimate of the treatment effect for each individual. So it's going to be 4 times AI minus half. So AI is either 0 or 1. So it's AI minus half times EI. That's what they can compute. And this is not shown to the explorer. And then this set R0 initially, but RT at the teeth step, is going to be divided into 2 parts, R plus and R minus. Those for whom delta hat is positive and those for whom delta hat is negative. So I just split it into 2 sets. And this is done to construct the estimate of the FDR. Okay, so I'm going to tell you what the rest of the algorithm is. But before I continue with the rest of the algorithm, let me just tell you a little bit more about delta hat. Like, why is this a reasonable estimate of the treatment effect or so on? Okay, so delta hat is the same formula that I had before in the previous slide. The key part is that y hat does not use the AIs, but delta hat uses the AIs. So it's reasonable because this actually, if you look at this estimator, it's actually used in even recent papers like by Nienwager and by Kennedy and so on. And it's like an estimate of the conditional treatment effect in some sense, but this actually can be traced back to much older papers by Robinson in 1988. But just for intuition, you can see that it recovers the true effect in simple cases. So suppose the yi control was C and yi treatment was C plus delta, and suppose y hat was just perfectly learned, and so y hat of xi is actually going to be C plus delta over 2 because I'm kind of averaging over the AIs and I'm going to get half treated, half controlled. So I'm going to get my expected outcome y hat is C plus delta over 2. And if you plug that in, then delta hat I actually corresponds to little delta, which is the the treatment effect in this case. So at least just for intuition, this delta hat I is a reasonable thing. But what we're really going to need is that for error control, it's not so hard to see that under the null, if the null is actually true, then the probability that delta hat I is positive, given all of the covariates and outcomes, is actually at most a half. Okay, so this delta hat I has this sign flip property. Those who are familiar with knockoffs or things of that kind, or masking of adapt, things of that kind, will already notice the sign flip property. But now we have managed to get this, like a bit, coin flip kind of property. We've managed to get it in a different setting of causal inference. Okay, so delta hat I is like a coin flip, and under the null, you can think about it as being equally likely to be positive or negative, or in fact more likely to be negative than positive in some sense under these 2 assumptions. Okay, I have a short proof, but I really won't go through it. And so, yeah, essentially the Y and X are being used to make selection events, but I get error control even conditional on that selection event because of this sign property. Okay, so now just to get back to the algorithm, so I compute Y hat, I give it to the Oracle, Oracle computes delta hat I for each person I, and divides my set RT into plus and the positives and negatives. And what Oracle tells back to the Explorer is the size of R plus T. R plus T is the set of people within RT that have a positive delta hat I. So it tells you how many people have positive delta hat I, and the statistician can then compute this estimate of the false discovery proportion. Okay, so it's just RT minus divided by RT plus essentially, and again very knockoff reminiscent, and they can just check if this is less than alpha. And if it is less than, again, yeah, this goes back to this knockoffs and adapt and several other papers, the inspiration for this FDR hat. And if the FDR hat is less than alpha, we stop and we just say, good, I can just report R tau plus as the people I think have a positive treatment effect. And if not, then actually I'm going to pick one person I star T, that is an element of RT, hopefully a null, that and has an estimated treatment effect less than 0. But remember, I don't know these estimated delta hat i's, so I have to pick I star T using whatever I know currently, and I kick that person out. Okay, so I basically, for that person, they're kicked out of the set, but I get to learn their treatment effect, their treatment assignment I star T. Okay, so this is where I'm learning more and more information. So I'm removing this person from the potential rejection set, but I'm learning their treatment assignment. And so now I can calculate that person's delta hat I star T and its sign, because now I know their AI, so I can calculate what actually happened. Did I kick out a potential person who was a treated person, or did I kick out a control person, like, in some sense? Did I kick out a person with a positive treatment effect or negative treatment effect according to my current model? And you can calculate that, and you can amend your model and so on. So then you update your new set RT plus 1 is just RT minus this person that you learned about. And now you send this back to the Oracle. You recompute your model. You can send these errors back and so on. So anyway, so these steps of 4 to 8 can just repeat, where you're constantly learning new information and so on. Yeah, so this just repeats until you're actually able to stop. So the way we got to that green set is we started from the full set, and then we one step at a time we removed people that a model thought was not going to have a treatment effect and so on. So the summary is I start off with some prior knowledge. I have a sequence of sets that are shrinking, and I stop if ever my estimate is less than alpha, and I report the set at that time. Otherwise, I unmask the treatment assignments AI. And so I have more and more information for selection as I go along. I'm learning more about the data set. And so the theorem is that under no further assumptions, but under just those standard causal assumptions I told you, just let's say it's uniform treatment assignment and no interference, this does control the false discovery rate for randomized experiments. There is an issue in the selection step. We would like to exclude subjects that have a negative delta hat I, given the revealed information. But at the very start, I have no treatment assignment, so I have kind of no information to choose who to exclude. So as just one example, if I know these xi, yi, the first two columns, some covariate information and the outcome, but I actually don't know the AIs, and the AIs could be the one in orange or the one in purple, and I actually have no idea. I have no way to tell initially which is which, and so I might end up kicking out a person with a positive treatment effect rather than with a negative treatment effect. So we cannot tell what the correct signs are for AI, and so this causes an issue with power. And so the fix to this is actually to wrap what I just told you, wrap iCube around a cross-fitting framework. So we call this cross-fit iCube. And so the idea is very simple. I'm going to randomly split the data into 2 parts, and I'm going to apply iCube on D1, where all the information in D2 is revealed to the analyst, meaning xi, yi, ai from D2 are all revealed to the analyst. But on D1, I only know xi, yi. And I run iCube at level alpha by 2. And then kind of separately or in parallel, I run iCube on D2, but I give D2 everything about D1, xi, yi, ai, but on D2, I only know xi, yi, and I run it again at level alpha by 2. And then the final rejection set is the union of R1 and R2. And so this, because I now have some actual information about how X, Y, and A are jointly, anyway, so this actually will reduce we'll get rid of the issue that I had earlier where I can actually determine from half the data which is the right assignment and so on. So now I know what's the right way to orient these people, like who's treated and who's controlled, because I have some of that information. So subjects with the same covariates are more likely to have effects in the same direction. And based on the effect signs of the revealed samples, we could prefer the correct signs over the opposite signs for all the for the other subjects. Okay, so effectively, what's really happening in the final algorithm is that I actually reveal xi, yi in D1 and everything in D2, and I'm running the I-cubed algorithm around that. Okay, and so like, how do I actually choose who to exclude? Like, here's an example. I use the non-candidate subjects, those who have already been excluded, but I have their complete data. Remember that anybody who is not in the current potential rejection set, I have their entire data set. And I train a random forest classifier to predict whether their delta hat is positive or not. Okay, and then I apply that random forest classifier to new people, because on those new people I know y, x, and y minus y hat in some sense, and so I can predict for those people whether delta hat is positive or not. And hopefully, if my random forest classifier was pretty good, then I will identify a good guess of who to remove from the set. So the I star t is just going to end up being some kind of greedy choice of the person who has the least probability of being positive according to my model. But the point is, I can change this model in every round. So like somehow if I start to see that I'm picking out people with positive treatments, I can guess that, oh, something's wrong with my model. I can adjust my model in consecutive rounds and so on. So the way I choose this I star t can adapt and change. I don't have to pre-commit to a particular model when I don't have to pre-commit to random forest, for example, or something like that. Okay, so some simple experiments. We have let's say a three-dimensional setup, yi control and yi treatment. So the treatment has there's a delta of xi, but it's going to be heterogeneous. So for example, you'll have some people with a large effect size and a lot of people with a small effect size, and some have a positive and they have both negative effects. So they have a lot of people with small negative effects and a small number of people with large positive effects. And so we're just comparing CrossFit with something like, what if I fit a linear model, calculated p-values, and applied Benjamin-Hochberg or something like that? This would give you asymptotic FDR control if the linear assumption was true. But if the linear assumption is not true, it doesn't control FDR. So when you run this, you see that, well, in this case, the FDR control is not guaranteed. It doesn't work for linear BH, but CrossFit I-cube does control the FDR. And especially if you look at FDR, when I focus on the how many of the people with negative treatment effects were selected, like linear BH actually picks a lot of people with negative treatment effects, but we don't. But power is comparable, and as some treatment effects gets larger, we even have increased power. There's many more figures in the paper and many more simulations, but just one example to show that the method actually seems to be powerful and control reasonable notions of FDR that we would like to control, and so on. You don't really seem to pay too much relative to a more naive method like linear BH. So I'm almost out of time. So we have some extensions to observational studies. I won't go through them, but we have a doubly robust FDR control. So when either the propensity scores are well estimated or the outcome model is well estimated, this is the usual notion of double robustness. But now we have that for FDR control, which is just we usually have it for average treatment effect estimation and CLD or something of that kind. Here we have it for a different goal, which is FDR control. So I think that's interesting. So to summarize, CrossFit I-cube outputs a set of subjects whose effects are positive with a guaranteed FDR in randomized settings and a doubly robust FDR in observational settings. We can take covariates and prior knowledge into account in a flexible manner. And they can identify subjects even if they're not treated. The paper has other things. We relax the null to control for non-positive effects. We show extensions to paired samples and to subgroup identification. We actually have a real data analysis where we take it on, we use an ACIC challenge data set with 500 subjects, 22 covariates, 70% of them were treated. The average treatment effect is very small. but it turns out we can identify a few hundred individuals as having a positive treatment effect. But a big limitation is that these are entirely in-sample results. So among the people who participated in the trial, I can identify, some of those that are super responders, but this is not for new subjects. Okay, so that's a limitation. Anyway, so let me just thank my co-authors again and thank all of you, and I'd be happy to take questions.
Thank you very much. It was a great talk. Because we are running a bit out of time, we can take a couple of questions if there are. So there is one question from Asser. Asser, do you want to unmute?
Yeah, thanks so much for the great talk. I guess my question is sort of concerning your last bullet point. I mean, so as you mentioned, I mean, there are maybe some cases where you might be interested in doing inference on external populations. Like, do you have ideas about how to extend to this case? Like, is there some concentration argument where, like, if you pick some rectangle, like probably people in that rectangle in the future are likely to also have positive treatment effects? I don't know. What do you think?
I think that's plausible, but it would require assumptions on the conditional average treatment effect. So like, I feel like you need smoothness assumptions so like somehow this is internal validity. And if you want external validity, then you need assumptions but that's reasonable. If you're willing to make smoothness assumptions, probably the rectangle I've identified, will carry forward to people I've not seen there. But so I agree on that front. And but we haven't written that down, and maybe somebody who's interested in follow-up work could try and see whether this can actually lead to clean out of sample guarantees. Yeah.
Thank you.
There is one more question. Let me see the name. Sophie, yeah.
That's
thanks.
me. Thank you. That's a very good presentation. I wonder, like in clinical trials, other than just identify these patients, we're also interested in how we can use this information either for neighboring purpose or the design of the next trial. I wonder for this method, could you help to demonstrate a little bit on whether you can use this to help to identify the patient features which might contribute to this positive treatment effect or things like that? Is it possible?
I think I completely agree with the motivation of your question. We don't have anything provable at the moment, but if I was a data analyst, that's the natural thing I would do is for example, I would look at the box that we've identified and say, okay, if it's likely that these features are important and in these ranges and so on, and that could inform your next clinical trial, but like not in a way that I can write a theorem about, but certainly in a way I think that is going to be interpretable. Like I think I would definitely use this to identify not just the super responders that were part of my trial, but what do these super responders have in common and so on. But yeah, we don't have a theorem for that, but I think it clearly points in a direction for what a reasonable data analyst should do if they were planning a future experiment.
Okay. Which part of the method can help to identify these features? Is it on the estimation of Y or?
No, it's more about in the end, if you look at the people that were identified. So like ours, we're going to output a set of identified individuals that have a large have a positive treatment effect, and it's more about just analyzing, well, how is that subpopulation different from the rest of in this 2D case, of course, you visualize it and you say, well, they're kind of a box in the middle, and those are the covariates in those ranges. In a more high dimensional example, you could just do some follow-up heuristic analysis of how trying to identify the features that try to differentiate the identified population so it would be more of a cluster analysis or a two-sample test kind of thing, or something of that, or variable importance test for what differentiates these identified people from the rest. But I think the intuition should be relatively clear. I don't have a theorem for what's the right way to do it, though.
Yeah, that makes sense. Thank you so much.
Yeah, sure.
Great suggestion. Any other questions? Yeah. Well, thank you once again, and thanks all of the speakers. Maybe Dominic, you can wrap it up.
Yes, we're a little bit over time, so I'll keep it very brief. I just want to say big thank you to everybody who contributed to making this such an excellent seminar. I'm sharing some details of future BBS events. I hope to see many of you there. You can find the details on the website and the registration links. But otherwise, just want to thank all of you for attending and wish you all a very nice rest of the day. Thank you very much.
Thank you.